So I'm going to talk about chemical tools to study advanced glycation end products. So one expression I really liked from Baines, who is a pioneer in the field, is he explains why ages exist, and it's from one perspective the human body might be viewed as an extraordinary complex mixture of chemicals reacting in a low temperature, often with a 76-year cooking cycle. So that's what actually the human body really is. So ages I don't have to go into, we saw it all yesterday, how they are formed. I want to point out, because I know some people sometimes do tell me that they don't know the difference between glycation and glycosylation. They are very different processes. But I think the majority in the public is aware that glycosylation is an enzymatic process that is needed for the normal structure and function of proteins, and that glycation is not increased in aging, although work in the University of Ghent has shown that there are changes in the profile of N-glycans, but there is no increase or decrease. It's only a change in which sugars are incorporated in the different positions. So why glycation? And that's important, so that happens on the lysine arginine side chains. Some reports on cysteine and histidine in the literature, but only a few. It's less site-specific, so it's a pure accidental process. However, of course, the side chain must be accessible for the reactive species to react with. And there are also side chains from the neighboring amino acids can influence the properties, such as the pKa of the side chain, which can make the side chain more nucleophilic or electrophilic and thus more or less susceptible to glycation. They are formed inter- and extracellular. They are, as far as we know, non-enzymatic, and there is no energy thus needed to make them. There are no energy in the form of ATP, GTP, or any other form the body uses of energy. It's generally just one sugar molecule or one reactive molecule, in contrast to glycosylation, where you have these large complex trees in the N-glycans, for example. There is extensive modification, several types of rearrangements, several types of dehydration, oxidation reactions, so the structures are heavily modified, and they distort the normal structure and function and increase in aging and diabetes, as we will see. So here are some ages we saw yesterday as well. So these are just the ones that attach to one amino acid, and these are the crosslinks that attach to two. So what is the suggestive evidence that ages are linked with aging and disease? First of all, the levels of various ages increase with age. The higher age levels or the higher precursor levels have been found in various disease conditions. So you have found higher methylglyoxal levels, for example, in disease conditions. Ages are increased in diabetic patients, and diabetes is a risk factor for many chronic health problems, heart disease, Alzheimer's. As far as we know, ages are unregulated. I have to say, as was pointed out yesterday, there is an enzyme that you can take the Amadori product and remove the modification, and I will come back to another method of actually taking an advanced glycation end product and removing it, but I think William Baines already pointed that out yesterday. But they are not like most of the metabolic things where you have very tight regulation going on. And there is some suggestive pathophysiological mechanisms by which they may impair normal function. Think about binding to RAGE, which was already mentioned, which initiates pro-inflammatory signaling, and so on. So they are increased with age and in diabetes, and you can see it here for 6 different products, including crosslinks and advanced glycation end products. They all increase. So just to show that there are ways how they can. So it's not my point of my lecture to go prove that they are involved in aging, but I'm just wanting to take one example, which is changing the mechanical properties, just to illustrate. So I won't go into age or anything like that. And you can see which was also already pointed out, that with age there is this is glycated ex vivo, the tendons of a rat with glucose, and you see an increase in the amount of insoluble collagen. So that cannot be extracted by acid or by salts. And when you then do measurements on the mechanical properties, you can see that there is a clear difference between the tendons that had been glycated, exposed to glucose, or the ones that had been non-glycated. And this is for the maximum load you can put on it, but the same is true for many other things like the Young modulus changes, the flexibility changes, so a whole range of difficult mechanical properties have been measured and have been found to change. And so this is just for neutral salt collagen extracted, acid extracted collagen. This is what has been told about yesterday. That's the peptides extracted, and then here the insoluble fraction, where you can see the changes opposite to the other ones. Because, so here you can actually see that this fraction, as you know, when you cross-link it and modify it, it actually becomes more insoluble. This insoluble fraction is the one that contains most of these cross-linking agents, and thus this is also the one that is more stiffer, and that is less flexible, that has a higher maximum load and so on. And here you can see they actually correlated the maximum load of non-glycated glycated with the levels of pentosidine present. Another study, they came to the same results. Basically, they used muscle that they took from mice that was in vivo aged versus young animals. This is just recently published. And so, using some assumptions, actually, they said that 73% was of the increase in muscle stiffness was due to changes in the ECM, and 27% to the muscles. But that was based on some assumptions they made. But it can clearly be shown that the advanced glycation end product levels increase. So what is now the benefit of using chemical approaches to study advanced glycation end products? The classical method, named chicken soup by one of the attendants, is a heterogeneous mixture, so also called H protein. Purification is complex. It involves several steps: desalting, pre-concentration, semi-pret HPLC, and achieving of stereo purity is almost impossible. So glucosapine, for example, has 8 stereoisomers, and using even HPLC, you cannot obtain one single stereoisomer. In fact, you get 4 peaks, which are probably 4 diastereomer isomer pairs. With chemical synthesis, the glucosapine shown on the image by William Baines yesterday, Christian holding the flask, that was 1 stereoisomer, so perfectly pure product. The classical method is prone to contamination with bioactive impurities. Obviously, amino acids and glucose together, bacteria love it. While in the chemical method, that's not the case. And the concentration, what concentration does a mixture have? If you could measure the concentrations of each of the different components in that mixture, you could put a measurement on each of them, but then you couldn't vary each and every one of them separately. So if you dilute it, you dilute every one of them. So you couldn't do real studies where you look at the effect of changing the concentration of one single H product. So that's also problematic in this approach, which is not the case here. And if you don't purify it up, because purification is really hard or almost impossible to do, and we will see some examples later, then you can't really make specific antibodies. And that's what William Baines said: all the antibodies, or almost all the antibodies that have been used so far, are really binding to everything. So pure chemical synthesis has by now achieved the synthesis of these compounds that you see here. So first of all, I want to go to something else. Methyl glyoxal, which is a precursor, so it leads to the methyl glyoxal derived hydroimidazolones. One of the, so that there were 3 of them, and they have all been synthesized chemically. So first of all, we wanted to essentially make a chemical sensor to measure how much methyl glyoxal is present in vivo. This is the sensor, and when you react it with methyl glyoxal, it gets fluorescent. The sensor is EMBO, that's the name, and it's specific. So here you can see fluorescence for methyl glyoxal, glyoxal, and a whole range of other dicarbonyls, and hydrogen peroxide as well, and this is with nothing blank. And you can see that methyl glyoxal is the only one where you have an increase in fluorescence, well, except for this side thing, but this is only present at very, very low concentrations in vivo. So when you see here methyl glyoxal and glyoxal, the only difference is one methyl group, yet the sensor is so specific you can actually see one methyl group difference. And so here you can see some cells that have been treated with 0, 5, 10, 50 micromolar, and you can see fluorescence increasing. And here this is this side reaction that also happens, and you see a very small amount of fluorescence. So that is present in vivo, and basically because it's so weak, it doesn't really interfere with the sensing of methyl glyoxal. Other chemical sensors for aging research have also been developed to measure reactive oxygen species, for example. They can be specific to the mitochondria by attaching this mitochondrial targeting structure to it, or for other things like the cell nucleus and other organelles. So that has all been developed. And so then the synthesis of molten gold, our lab was not involved in that. So those were people from New Zealand, and basically they take an amino acid that has an FMOC protected group, and they add glyoxal or methyl glyoxal. They react it at 50 degrees for 3 hours, and you get this. And then you can, if you want, cut this off, or you can use this for solid phase peptide synthesis, and they proven you can make peptides with incorporation of the molten gold in the peptide. So what our lab then did was developing of these MGHs, and the really interesting one was the MGH3 here. So in low pH, you get just protonation, nothing happens. At high pH, it degrades into carboxyethyl arginine, which is in equilibrium with MGH1. However, then, if you take this at neutral pH, in the presence of reactive oxygen species, and we have only done this in pure chemistry, we haven't tried if this really happens in vivo. But in pure chemical circumstances, if you add an antioxidant to the mixture, you will get MgI3, methyl glyoxal imidazole, number 3, which spontaneously releases pyruvate, which is a metabolite, and you get arginine from it. So basically here you see how one of the H products, which have always been said that are irreversible, can actually, upon oxidation, spontaneously decompose and form back the native arginine. So interesting from that is, of course, you saw ROS, so they basically is an antioxidant. And it has been shown in different assays, MTT, DPPH assay, that basically MGH3 works as well as ascorbic acid as an antioxidant. So then one other project that was also already mentioned was the use of ELT711, also called allybrium chloride, to cleave dicarbonyls into non-reactive aldehydes and acids. The classical synthesis and purification method for pentosylin has been published, again using, in this case, Boc-protected amino acids and ribose for 6 days at 65 degrees, pH 8.5. You get pentosylin that has Boc protection, and then with TFA, that's an acid you can cleave off the Boc-protected group, and you get pentosylin with an overall yield of only 0.83%. Now, this extremely low yield means if you want to make any material for studying, you will have to make a lot of this pentosylin, and purification with HPLC of that amount of material is going to take you quite a while. In fact, a colleague of mine had to purify 1 gram of material for a completely different thing. 1 gram, and he took a week on HPLC to purify 1 gram. So imagine if you have to do it on hundreds of grams or so to get any pentosylin. A microwave-assisted synthesis has been developed, again, not by us, by others, and there you get up to 8% yield. The total synthesis has actually been published already in 2001. The problem is it's 17 steps long, so it's a very complex thing. It will also take you a few months and a very good organic chemist to make it. And then, of course, all 17 steps are not shown here. This is only the last steps, actually. So finally, what we have been doing is trying to make glucosamine, because the evidence suggests that glucosamine is the most relevant, or the most abundant, crosslink in vivo. And so we have started with D-aceton D-glucose, which is this material, which is commercially available and it's cheap. And in 3 steps we can come to an intermediate that we hope, but this hasn't been done, we have done some trials and basically it didn't work out. So we might revisit that in the future, we don't know yet, but we hoped we could make pentosynane, which is a precursor of pentosidine. Now the problem is that nobody really knows which of these two are really present in vivo. The thing is, of course, that when you do a measurement, you take a sample, for example, a biopsy, you need to degrade the collagen, you need to split it up, you need to then remove fat, do all these processing steps before you can do any useful measurement on them. And during those processing steps, you may actually cause the formation of pentosidine from pentosynane, if pentosynane is the agent that is present. So due to that limitation, we really don't know which of these two is really present in vivo. And there is discussion about it based on pure chemistry, which one of them is the most likely one to be really present. So we would hope if we could make this, we could actually use it as a tool to study which of the two is present in vivo. And so using this common intermediate in 7 steps, we can make glucosamine. Well, the number of steps is really dependent if you want to make it like this, or with protective groups on it, or if you want to make a peptide, then it's of course many more steps, and we haven't done that yet. What are the future perspectives? Designing selective antibodies is really important. Then better understanding the tissue, the age, and the disease differences in age levels. Ideally, you would, in one condition, for example, one set of mice, measure all of the different ages, all of the different crosslinks in various sets of tissues, and then have a really better understanding of which ones are the most abundant ones, and in which tissues they are the most abundant. Investigating the binding of these ages to cell surface receptors, because most of the evidence to date has been done with what has been called chicken soup, which binds to everything, and so we really don't know for certain if these ages really bind to the various receptors that have been suggested they bind to. Investigating the physiological relevance of metal-ion binding, so for example, iron can bind to some of the advanced glycation end products and may be involved in generating oxidative stress. So that's something that is not well understood at this moment. Investigating the biomechanical impacts of extracellular matrix protein cross-linking. So what does it do with tissue stiffness and how is that involved, for example, in the cardiovascular system. The possible role of age modifications in normal physiological processes, like I say, do they act as an antioxidant? Are they reversible? And then investigating the reversibility of these age products on the physiological concentrations. And of course, last but not least, looking at if they are involved in aging, then we really need to look at therapeutics and crosslink breakers. And the acknowledgements, so the PI of the lab was David Spiegel. The crosslinks research has been done by Christian Rahisi, who showed glucosamine sample yesterday on the slide. And the graduate students Kenneth Kahn and Matthew Streeter, who joined the lab last year. And the methyl glyoxal research has been done by Tina Wang, Eugene Douglas, Kelly Fitzgerald, Ethan Sanosi, Randy Kartika, who has graduated since and become a professor of his own lab. And finally, the ALT research was done by Phil. And the Sense Foundation provided funding. Thank you very much.